Good morning, everyone. All right, recording in progress. Good morning, everyone. I'm very happy to see some number of you here and some other number online on this beautiful, sunny, ice cold day. And I'm happy to introduce to you Jeremy Mulich from here on the quad, on the school. He is a director of software engineering development at the LSP and has been here, well, with short breaks on the quad since 2002, three, one. First for a couple of years, then moved to the MIT to join Peter Sorger's lab, and then Peter Sorger came back here. So since then, he's been here on the quad, doing great work. Some of the work that I hope will be covered today is going to be Ashlar and MC Micro and other lovely software tools that, though I'm pretty new here, I was surprised to find that I knew about them and people that came to me for advice for image analysis did not know about them. I hope this talk will sort of remedy that a little bit, but otherwise we'll just keep on telling people about it. So Jeremy, please.
Thank you, Simon, and thanks to CCP for inviting me. Simon said, so I am the Director of Software Engineering at the Lab of Systems Pharmacology here at HMS. So for those of you who aren't familiar with LSP, LSP is a multi-PI collaborative team science lab. And so our approach, we use team science to advance the fundamental science and technology needed to develop new medicines, personalized patient care. We study the molecular causes of disease, how drugs exert their therapeutic and adverse effects, how clinical trials are designed and interpreted, and how machine learning and AI can help identify new molecular targets in diseases like cancer, Alzheimer's, tuberculosis, and other infectious diseases. So what do I do at LSP? So like I said, I'm the Director of Software Engineering, and I directly develop on and help support through my team a series of internally developed software tools to help manage data processing, visualization, and publication of large data sets that we collect, as well as training members of the lab in the use of the software, in programming in general. We try to run software carpentry for Python. I've fallen behind in getting it scheduled since the pandemic, but that's something we're going to start doing again. Once we get through our backlog of folks in the lab who want to take the course, we'll open it back up and do a more sort of broad audience Python training course. I know that there's some Python courses here from other groups at HMS, but I think they're like a couple hours, sort of an intro course. This is really like a 2-day intensive. If you've ever taken a software carpentry course, you know it is intensive, hands-on lab coding. So that's fun. So again, back to my title. So it's a bit of a mouthful. Image processing, visual analytics, and data sharing for whole slide imaging and spatial profiling of tissues and tumors. So I'm going to kind of work backwards and unpack that and sort of work backwards into the meat at the front of the title there. So why spatial tissue and spatial analysis of tissues and tumors? We have in medicine over 100 years of histopathology of expert medical practitioners looking down an old classic microscope at thin sections of tissue stained with generally hematoxin eosin to visualize nuclei and certain other tissue morphology tissues and cellular components. And through just a lot of training and expertise and the power of the human brain, can diagnose and identify a bewildering array of diseases and disorders, and also extreme subtlety in cellular and tissue morphology and spatial patterns. We're just recently, I think, starting to get to where we can measure this kind of or measure and analyze this kind of pattern computationally. But we always, in our group, we always refer back to our expert board certified pathologists to help us understand what we're actually looking at in a piece of tissue that we're analyzing, and to propose new techniques and new approaches in how we might develop more powerful analyses in the future. So sort of taking that as sort of the basis for how we go or want to explore human disease. I think this is from a review last year of various spatial tissue. I think 2020 Nature Methods claimed spatial tissue profiling as the method of the year or whatever. Since then, the number of methods and instruments and techniques has only grown. This is sort of a hard to read table summarizing the different techniques, but basically a lot of them are RNA based. Everybody loves sequencing. The methods on the left are protein based. This is the one that sort of the protein methods are the ones that have always caught our eye because this is more akin to that H&E staining where you're looking at actual tissue morphology. And while I think the RNA methods have their place, we've sort of made our bed and we've gone all in on protein-based tissue imaging. So one of the best parts about using a microscope to do this is you're only limited by, in theory, diffraction limit of the light source you're using. You can get lots of different types of instruments. They're relatively affordable. I mean, microscopes can get expensive, but they're pretty common to have around. So you have something that's, you don't have to buy too much special equipment. The antibody cost can be high. That is one challenge. But a lot of the other variables in this table sort of, other than those, fall pretty well in favor of antibody-based immunofluorescence tissue imaging. Classic immunofluorescence, you're only limited to the number of sort of reasonably distinct wavelengths you can image on your microscope setup in terms of filters and light sources. One thing that we've pioneered in our lab is a technique called SciSif, which uses a cyclic approach to restain your sample and reuse I don't know if that was a question, but if anybody does have a question, you can go ahead and we can do questions during the talk instead of at the end. So cyclic imaging takes the classic immunofluorescence where you would stain with a number of antibodies or the fluorescent dyes and image with a cyclic approach where you can bleach the fluorophores or in some cases strip the antibodies. We have developed an approach where you bleach the fluorophores with hydrogen peroxide and bright light. Now that your sample has no more fluorescent signal, you can go back in with another round of antibodies and do the same thing through multiple cycles, align and stitch the images on a counter stain. We always use a nuclear marker in every cycle to align everything on. So you've got a large piece of tissue that's bigger than your microscope field of view. So you've got to take lots of small overlapping fields of view and stitch those together, again, using that nuclear channel to align them. And then the different cycles of imaging have to be aligned to each other so that you've got, say, up to 60 channels of immunofluorescence signal aligned at the single cell level that you can then analyze with all the tools of computational image analysis and also the classic pathologist's eyeball. So again, the benefit of SciSif is you can image an entire glass slide, so classic histology slides. With up to 60 channels, really the limit is both how well the tissue holds together throughout this process. So there's a lot of washing and decover slipping, and tissues, depending on what kind of tissue they are, tend to fall apart after a certain number of cycles. You're also limited by how many good antibodies there are. So validating antibodies for this technique is a chore. We've gone through a lot of that. We try to make that information shared as publicly as much as possible. But finding the right antibodies that are compatible with the optical setup on your microscope and have epitopes that you're interested in imaging in your sample, 60 channels usually tends to be about the max. The images are very large. So when you're imaging a whole slide image at, say, a couple centimeters across at 0.3 or 0.6 microns per pixel across 60 channels, you get 50,000 by 50,000 pixels by 60 channels and 16 bits per pixel. That gets to be a lot of data. So one of our other challenges is just managing all of that. And another nice benefit, as mentioned in the table on the other slide, was that it is FFPE, so formalin fixed paraffin embedded, which is the standard preparation for histology slides in the clinic. It is compatible with that. There are a lot of techniques that aren't for various reasons. I think RNA sequencing is famously generally not super compatible with FFP. I know there are techniques that do make it work, but the proteins come out perfectly fine.
Question,
yes.
So you say up to 60 channels. Is that like the top speed of a car, meaning you never reach it? What do you do sort of in daily life?
In daily life, I would say I see at least 30 to 40 channels a lot, but then it's a question of how much of the tissue falls apart. Sometimes the parts of the tissue that fall apart, the less cellularized parts, areas around necrotic regions, maybe adipocytes, maybe you didn't care about that part, but maybe the core that the tumor tends to be much denser does hold together. But I see 60 channel images around the lab pretty frequently, so it is achievable. So again, that's 20 channels. And then, so let me think about that. 60 channels, I guess that's probably a little bit that's on the high end. That's, I think, on the extreme end.
Just to follow up to this one. So, so the main limit on the number of channels is tissue integrity?
I would say so.
Okay. Okay.
But it is definitely the case that depending on your biology, what you're studying, available useful antibodies, you start to run out of those too.
And is it then that measurements of like later channels are kind of like worse and you need somehow like normalize for?
We absolutely do. I'll mention this a little bit briefly later, a tool to manage this. Optimizing the panel design is critical. We definitely want to put the channels, the antibodies for the proteins you care about in the earlier cycles when the tissue is less likely to have fallen apart. There's also a trick that in the very first cycle, you can use a primary secondary antibody combination. After that, you can't, because we don't wash the antibodies off, the primaries are still there. So you can't use, those, the IgGs and everything are all there. So you can't come back in with secondaries because they would rebind what's already there. Yeah, if there are questions on antibody selection and priority. Great. So, like I said, these are 100 gigabytes. It's 50 to 100,000 pixels across, 60 channels. You can do some things looking at it by eye, and again, the pathologists that have seen this love looking at the images because they can see sort of molecular detail at a subcellular level that they have only sort of imagined through the H&E stains. We still want to do computational, rigorous reproducible computational analysis on these images. So there was really, when we started doing this, there was nothing out there that could handle data this big. And tissue images in particular were not handled very well by a lot of the standard analysis packages out there. So we set out to build something. And so the pipeline that we ended up building is called MC Micro. Stands for multiple choice microscopy. I'll explain the multiple choice in a bit. So this is a sort of flow chart of the different modules in the pipeline. And so we start at the top with a whole slide, glass slide. We scan it through one of these various cyclic high-plex technologies, SciSif. It's also compatible with Codex, multiplex immunofluorescence, CellDive. Again, that table had 7 or 8 others that have sprouted up since the same type that have sprouted up since we made this figure. And then those raw image tiles. So again, you've got a field of view that's much small from your microscope, is much smaller than your tissue sample. So you've got to take a bunch of overlapping tiles across, sort of overlapping in X and Y across your entire sample. And following that, the first thing we do is illumination correction. Sorry, one other thing I want to mention is, so I go back here. Before I get into the modules, actually, I should talk a little bit. The overall orchestration of the different modules it uses the workflow language Nextflow. It's a nice, powerful, sort of pretty modern workflow language, and it can use Docker containers for the individual modules. So this helps solve the problem of setting up various tools on the computer you're running it on, whether it's your own computer or a cluster. And we've run this with various different types of imaging data, including some of these non-fluorescence techniques like imaging mass cytometry and MIBI. So the big news since the last time we've talked about this at this group is we are moving to NF-Core. So NF-Core is a community of best practices around Nextflow. So Nextflow was sort of a general purpose programming language for pipelines. There's a lot of different ways to do things and a lot of different ways to organize your documentation and your parameters and everything. NF-Core is a community started by some of the Nextflow developers and other folks, especially in bioinformatics, to standardize on those practices and form a community where documentation and sort of core code repositories and testing and all these things are standardized. So that once you learn how to run one NF-Core pipeline, you can transfer a lot of that knowledge to the other pipeline. Obviously not the scientific application itself, but it's a lot easier than with arbitrary next flow pipelines. So something that's in progress right now. I'm going to talk a little bit about some of the actual modules in here. So the first one is illumination correction. So we are shamelessly borrowed and stole good. So I said there was a lot of things that didn't work well, or so that we didn't scale to whole tissue images, whole slide tissue images. However, things that worked, we were happy to incorporate those. So one great example of that is BASIC. As you can see an example on the left, there's a it's a 2-channel image with about maybe 6 by 4 tiles, and you can see the grid lines showing the sort of tiles themselves. They're aligned perfectly well. The problem here is that because of optical realities of the microscope, the outer edge of your image is usually dimmer. Luckily, this is deterministic and can be modeled. You can either acquire a physical image to serve as a correction image to undo this dimming effect, or several tools such as this one called BASIC, given enough images with enough within certain patterns of variation that are required in the image, sort of equal probability of foreground and background, for example, or cell and not cell, can statistically extract that pattern. And then if you look on the image on the right, you can see what the image looks like after it's been corrected. So obviously for visual purposes, you don't want that grid, but also if you're quantifying the intensity of, say, the red and the green marker in this case, you wouldn't want to mistake a cell in the corner of a field for being low in expression when it really was just, it got less light. Simon?
So a small technical question. I know there was another tool out a few years earlier called CIDR, and I think you also considered that.
Yeah, we tested that, didn't work as well.
All right, because it needs more data, or because I thought basically theoretically you're supposed to perform better, but maybe only in the thermodynamic limit of whatever, a gazillion images.
We tried it and just it didn't. The correction profiles just and the images we got out just didn't look as good. Just empirically, I don't have anything better than that. So we've incorporated that as a standard module. So your raw tiles get fed into BASIC. This can all be parallelized because if you've got 10, 20, 30 cycles of imaging, those can all be processed in parallel. So that's nice. That's one thing that Nextflow helps you do is, if you have a process that can be parallelized, you can sort of split all your input data, run all that tool in parallel, and then join the results back together. So Simon also mentioned Ashlar. So this is the stitching and registration tool we wrote. So we found that existing image stitching tools and existing image registration tools both worked fine, but generally didn't scale to enormous images, just in terms of memory footprint especially. But the other problem we found was that, so let me give you a little bit of a schematic of a SciSif, sort of the stitching and registration challenge of a SciSif image. So in the sort of blue grid there, you can see representative of a DNA channel, tiled, so you have a 3 by 3 grid of tiles taken across a sample, say a small sample, and they overlap in the dark blue regions. And that's the area that you would use to align neighboring tiles to produce an aligned image. And then there are multiple cycles of imaging. Here 3 are shown. And then in the red box labeled C, you can see that the successive images of the same part of the tissue have to be aligned to each other, so that when you image the same cell with your other cycles of markers, the signal can be aligned and become one, are aligned on the same cell as soon as you analyze it. All the signals in the right boundary. And the goal of this is to produce one single image that has, say, the width of your sample by the height of your sample by the number of channels in your image in one large image. We can then split that apart for further processing in parallel, but we found that this was key because if you tried, so one thing that people propose is, well, what if you just took this, say the upper left tile from all the cycles as a, say, a 60 channel image and analyze that as a 60 channel image, and then do the same thing for the next tile over and the next tile over. The problem we found is that, well, this works for a single channel or for a single cycle. Once you have multiple cycles, every time you put the slide back on the microscope, either the stage doesn't necessarily go back to the same position every time. If you tell it to go to 100, 200, it might be off by a few microns. Also, if your microscope doesn't have a sort of super rigid slide holder, sometimes the slide slides around a little bit by more than a few microns. And so when you go back to those different rounds of imaging, the same tile, say tile number 1, is not always that close to where you started. And so you can have some shifts on the edges. Some of your signals on your later channels will be kind of pushed off to the side, and sort of you're going to lose some cells on the edge because you won't have all the signals from all the channels present on that tile. So what we decided to fix that is stitch first and then register. But the trick is, so this is just a depiction of what stitching looks like. So here is an intersection of 4 neighboring tiles and the DNA channel shown just in 4 pseudocolors. You can see a couple triangles indicating cells that don't align very well. And then after stitching, you can see that they line up very well and represent as one cell. And then registration is the same idea. You can see 3 different cycles. The same cell indicated in each doesn't line up. After registration, they do. So the trick we realized is we also can't stitch the entire image first and then stitch each cycle by itself and then register those images, because any small errors or inconsistencies in how the stitching is performed across an image where you've got, say, 30 tiles across, each one of those 30 alignments is a chance for slight error. So if you try to stitch 30 tiles by 30 tiles, and then do the same thing for another cycle, and then try to align those 2 images like this to each other, any of those little mistakes, you can't recover them. So once you've committed those stitching choices to an image, it's basically you've got something that's slightly wrinkled, or sort of images that are sort of slightly wrinkled with respect to each other, and there isn't any affine or any reasonable transformation that can recover them. And furthermore, if you've got 2 nuclei that are slightly non-overlapping, or the same nuclei from 2 different tiles that don't quite touch each other, you can't go back and correct that later, even if you got it right in another cycle. So our approach is stitch the first cycle, and then for every tile and every other cycle, register the corresponding cycle, or sorry, register the corresponding tile to its nearest neighbor in the first cycle. So this has the effect of sort of controlling the error propagation across through the stitching in the first cycle and through the registration to the other cycles. And works very well. And I'll show some examples of that now. So the way we thought, how to sort of prove this, how do we demonstrate this, is we took a 2-cycle data set and fed the tiles into the best open source stitching tool we could find at the time called MIST, which is provided as an ImageJ plugin. And we fed cycle 1 and cycle 2 to MIST separately, to sort of what I just said doesn't work too well, and produced 2 separate stitched images. And this is an example of a colon, a healthy colon section. And then compare, I'll explain in a minute, how well those 2 things lined up to each other. Same thing, fed it through Ashlar, using its all-in-one approach, and compare how well those images line up to each other. This is a bit much, but I'll explain what these colors represent. So the way we decided how well those 2 images lined up to each other to see how accurate or consistent the stitching was is through optical flow. But then we found out that all the standard optical flow algorithms crashed or used too much memory when you tried to run them on a whole slide image. So we came up with an approach to sort of coarse block-based optical flow. So we took 200 by 200 pixel image, 200 by 200 pixel patches, which is about the size of the inset panels you see on the bottom, and then walked that 200 pixel window across the image, did a local rigid registration, and said by what distance and what angle are those 2 patches shifted relative to each other, which is optical flow. Presented that on top of the image as a colored heat map, where the hue represents the angle, so red is to the right, green is down to the left, and so on, and the intensity of the color represents the distance. On the left is the example from MIST, where it was a tool that doesn't know anything about cyclic imaging. We just stitched the 2 cycles and aligned them, and this is what you get. You can see that there's uncorrelated errors, uncorrelated sort of differences in the way the images are aligned, the tiles are aligned all the way across the sample. The yellow X's indicate where those 4 insets are taken from. On the right, we have Ashlar's approach, where you can see there basically is almost no color. So this means you can see the insets on the bottom. The 2 cycles are aligned almost perfectly everywhere through the image. There are a few sort of dustings of color. This is places where the tissue fell off the slide or was not well attached and sort of flopped around uncontrollably between the 2 imaging cycles. Basically something that's uncorrectable, and that's That's fine. It's something we part of the sample we would end up discarding regardless. And then if you quantify the KDE plot of the distance of those different shifts, you can see that you can see in another way, which is maybe more clear cut, that Ashlar performs much better. We also had a fun, we also had a fun time when submitting the paper, and one of the authors, clearly not understanding microscopy, said, "Don't slide scanners just solve this problem?" So, well, first of all, a slide scanner is just a microscope with some software attached to it that does stitching in sort of internally, so it's not magic. And we said, well, how good are the slide scanners? So the classic histology slide scanners used in the clinic or in research. And the approach we came up there was we took one slide and scanned it twice on the same scanner, and then treated those as though they were 2 cycles. But since the sample didn't move at all, this should be very easy, so that the sample hasn't been cover slipped and washed, and the tissue shouldn't have moved around at all. So this is kind of this is the best case scenario for any algorithm that's going to be stitching an image. If it does it well and consistently, this should be easy. So then we took the same slide, scanned it twice on a research microscope that can do white light imaging, and then ran that through Ashlar, did the same thing. Here's the result. In the upper left-hand corner, you can see an H&E image just for sort of reference and scale about how big the sample is. In the upper left, you can see the results using the optical flow method from Ashlar. There is a little bit of pink and blue. I think that's a spot where the autofocus microscope didn't do great, and so those tiles weren't super aligned. And then you can see 3 successive commercial bright field slide scanners. In the upper right is the Leica GT450. This is actually really good. I was really impressed by this. You can see a little bit of colored banding, but it's not too bad. The bottom left is the Leica Aperio Versa. This has a very different pattern of squares. And then on the right, bottom right, you've got the Hamamatsu Nano Zoomer with some other weird stripes. So this is weird until we looked into the actual mechanism by which these microscopes were acquiring images. The Leica GT450 and the Nano Zoomer use a line camera, not a rectangular sensor. So they use like a 4,000 pixel line camera that they sweep across the image. And then they move the stage over, they sweep the stage over the sensor, and then it moves the stage over and swipes. And so basically it takes swaths like this. And you can actually see that pattern in the error field. The Versa has a immunofluorescence option. There's a fluorescence option. So it has to have an area sensor so that it can take an image, change the light source or the filters, take another image, take another image, and then move to the next tile. So that's why it has that sort of farm field grid approach. So we thought that was interesting that, and then you can see in the bottom left the quantification of the error. We thought that was a really interesting sort of insight into the construction of these microscopes that you can see just by their sort of accuracy of their software stitching. Next, and I apologize, I didn't get a chance to add this to the figure, but background subtraction is something we've added recently by popular demand. Previously, we've tried to handle the illumination correction well enough that background is pretty even across the entire image, and subtracting a fixed value solves it. But in some cases, people have found that they do need some more advanced background subtraction. So we have 2 modules, one of which just takes pixel-wise subtraction of a reference image. So some people will pre-image their sample with no antibodies or with control antibodies to get an autofluorescence or control image, use that for subtraction. And other people like to use rolling ball, so we just added a module that calls ImageJ rolling ball. Again, happy to beg, borrow, and steal stuff that works. Next thing is if you are imaging a tissue microarray where you've got lots of small samples from different patients arrayed in one slide, people generally want to analyze each of these separately. So we have a tool next to Clarence Yap called Choreograph. That's a UNet-based trained model that will detect where the cores are in the TMA, crop them out, save them in separate images, and then the rest of the processing downstream can actually happen in parallel. One thing is we've found that TMAs, sort of as an aside, the spatial distribution, sort of the limited spatial sampling you get by taking a 1 to 2 millimeter core of a tumor is wildly different or very biased compared to what you get from the entire tumor. So we have a paper demonstrating this. So if you are interested in TMAs for tissues, be careful about what questions you're asking because you may not be sampling the biology in the tumor that you think you're sampling. And then after we've aligned and stitched and potentially split the TMA cores, we go on to segmentation. So this is the case where we want to identify the boundary of all the cells in the image so that we can then do further analyses and quantification on those cells. This is where the sort of multi-choice in MC Micro comes into play. There were not a lot of great options when we started. So again, we wrote our own. So Clarence Yap was the lead author on the segmentation tool called Unmixed. It's a UNet-based deep learning algorithm. And his insight was that just using a DNA channel and some augmentation.
Jeremy, just one question on the slide before, but these four, can you talk a little bit to when you how you decide between these four based on use cases?
It's just personal taste. People either have used one before and they like it, or they can run all of them and look at the results. And sometimes, depending on the training or just various details of the algorithm, they can get better results on some tissues.
Okay.
So we lead them all in because this is one of the unsolved problems right now in tissue imaging is segmenting tissues.
And what's your empirical impression? They all work about the same?
They all have their strengths and weaknesses.
Gotcha.
Yeah. One of the strengths, for example, with Elastic is they have a really easy desktop application workflow for training on your own samples. So a lot of people that have challenging tissues, we've found they'll take Elastic and they'll sit, outlining some cells for an afternoon and get pretty good results out of that. Whereas training some of these other models you need a lot more setup and infrastructure to get it going.
And do you have experience with this segment anything for microscopy, Sam?
We took one quick shot at it when it was first published and didn't get good results, but I've seen other groups have better results with it since then. So it's something we do want to look into. Yeah, so there's the ones that we've got. Again, if you are interested in using this pipeline, there's an algorithm that you are interested in that's not here and you think it works well for you, let us know. We can look at adding it in. It's a pretty easy job. The one thing I want to just give a shout out to Clarence, the thing that he realized is that the classic augmentation method of adding more synthetic data to your data set when you're training a segmentation algorithm, which is to blur the images so that you simulate data that's actually out of focus a little bit, so you can sort of recover slightly out of focus signals. He had the idea, what if we actually take physically blurred samples and we'll actually drive the Z stage of the microscope up and down by 3 microns for every image that we take, have the humans annotate the focused image, but then use the physically blurred data in the training set. That actually worked a lot better than this just Gaussian blur your image. So for whatever reason, using the actual OTF of the microscope actually gave better results than just a sort of a fake one. Who knew? Also adding a nuclear envelope stain helped a lot. So a cocktail of laminin antibodies and a few other things helped identify, no surprise, the boundary of the nucleus a lot better. This is just showing the performance, the intersection over union performance of that approach. So now that we've got segmentation boundaries for all the cells. Oh, one thing I should say is that another challenge here is that nuclear segmentation, because we've got a DNA stain everywhere, every cell almost has DNA. Segmenting the nucleus is achievable. Segmenting the cytoplasm is a lot harder, or other organelles, because there isn't one universal plasma membrane marker for all cells. The best we can do is find as much coverage as we can for the cells we care about and add them to the experiment, but it can be tricky. Adding them all to the same cycle can be hard because you've got to get the dilutions correct so that you get roughly the same intensity. Otherwise, you have to use multiple channels and multiple cycles to use them to add those all. So for a lot of times, we actually fall back on just dilating a fixed number of pixels from the nuclear mask, avoiding not overlapping neighboring cells, and calling that good enough. We would love to have better full cytoplasm segmentation, but I'll talk about something in a minute that can maybe start to work around that a bit. So after we've got segmented cell masks and the original image data, we can perform quantification and various other downstream analyses. Quantification right now is a very straightforward scikit image region props call where we just take all of the cell masks and then quantify some basic morphological features and the average intensity of all the 60, however many channel markers you've got within the masks of each cell. Produces a CSV file, and that's our quantification table. We've thought about running more of the cell profiler approach of measure as many properties as you possibly can, and then just sort it out later. These files with 30 to 60 channels are already getting really big, and so we might want to be a bit more thought about this, but we should probably be more judicious about what markers are useful. I know that kind of defeats the purpose of, I saw Beth's talk the other week, and I've seen Anne talk many times about this approach of just measure everything and sort it out. But the data size does actually really start to hit you if you start to do that on these images. So after we've got the quantification table, we've got to do something with it. And so there's a number of modules we have built in to do kind of a first, and this is all very much dependent on your biological question and your tissue. So there's nothing cut and dry. This is where the sort of everything diverges into your own bespoke needs for your tissue and your biology. But we do have a few first pass tools here that can do some clustering and other types of analysis to at least give you a first look at what your single cell data looks like. And I'll just call out especially SciMap, written by Ajit Nirmal in the lab, now his own lab at across the street. So quick diversion here is quality control. When you're imaging a few cells, and you're just looking for a nice, beautiful image in a dish, you can probably look around and find a field that looks pretty good. When you're scanning an entire slide, an entire tissue sample, and you want to get all the cells for, because every piece of the tissue is different, you don't want to miss any rare events. You can't pick and choose, or if you're doing 10 or 20 or 100 slides, you can't sit there and pick and choose parts of the tissue because they might have a fold or a wrinkle or some other problem. Also, there are things that only become evident after you've imaged, such as antibody aggregates, or you've got a hair or a piece of a fiber or something on the slide that's fluorescent. There could also be problems that happen only during the imaging, like a piece of the tissue detach, air bubble under your cover slip. In the bottom right, I don't know what happened there, but that's a mess, probably dirt on the objective or something. So there's a lot of things that can happen, and especially with precious samples. So we've done some work with very rare precious patient cohorts where we only got one crack at each of these samples. We can't go back and ask for another section from the sample because it may not exist, or too many people wanted access to it. So we found that we have to clean this up and do the best we can. So we're trying to develop automated methods, but there just isn't enough training data to go on, and it's still not sure kind of what the scope of the problem is. So for now, we've got a human-guided, computer-assisted tool. This tool by Greg Baker in the lab is called Silenter, and it has a bunch of sort of, for all the kinds of artifacts that you saw before, there are different statistical approaches and graphical plots and sort of ways to compare the single cell and image data that Greg came up with that he can, that the code presents to you, and the user interactively steps through the channels and the samples and makes their own call on what they think is artifact. They can draw boundaries to draw a region of interest around cells to exclude it. They can use intensity-based gating to filter out cells that are obviously way too bright or way too dim or way too big or small or whatever the metric is. And this is all based on Napari. So it's a series of plots and data visualizations in Napari that let you load your image, explore the single cell data, explore the QC plots, and filter your single cell data to exclude the artifacts. I think this is in bioRxiv, and I think the actual paper should be out anytime soon. It's just in final review now. So I mentioned before about the sort of work that goes into designing antibody panels and sort of managing, I didn't mention clinical data, but the other thing is clinical cohorts. So we're receiving slides from collaborators at the hospitals. They're busy, they've got a lot to do. They can't sort of manage this stuff for us. So we get a box of 10 or 20 or 100 slides. We've got to figure out what's what and keep it distinct from all the other projects, figure out what disease, what patient demographics are associated with the slides, so that when we go and analyze the data, we know, okay, these were the smokers and the non-smokers, or the progressors and non-progressors, or whatever the metric is that we're analyzing. So we built a couple different laboratory information management systems, one of which is called Reagent Tracker. There's a screenshot on the left. This was originally designed to handle our small molecules. We do a lot of small molecule profiling in cells. We've extended it to manage our antibodies as well. So when you're doing an experiment with 60 little vials of colorless liquid, pipetting them and sloshing them on your slides, you want to make sure you've got the right ones from the freezer and that you've got the right ones in front of you and that you decided to pull the right ones in the first place. So we've got a series of data curators and managers who enter information on these antibodies as soon as they come in. Things like the species and the target and the fluorophore and the vendor and the lot number and other quality control information that we've collected. And then that is used on the experiment tracker side, which is the other half of this. That experiment tracker manages sort of slide onboarding, clinical data collection, development of the staining plan. So we've entered information about our different microscopes and their filter sets, so you can decide which fluorophores are compatible with which channel and which cycle in your experiment and put together an antibody panel that works for your instrument in your biology. And then this system also manages sort of project slide IDs and project IDs that we use to name folders and files on disk, because when you've got this many files and this many projects going on at the same time, we need a reliable, rigid naming scheme. That's that. I'm going to point out something that people may have noticed here is that this workflow feels kind of similar to RNA-seq, where you start off with some raw reads, you align them into longer reads, you quantify those per sample, whether it's single cell or what, and then you've got a counts table of, say, cells by genes. Our workflow is kind of similar. You start with raw tiles, you align them to each other, you quantify them down to the single cell level, then you quantify the counts of some protein concentration in each of those cells. This seems desirable. Genomics kind of sucks all the air out of the room, especially compared to microscopy, I think. And we want to be able to use all the cool tools that genomics is building for clustering and analysis. And so this seems like a good thing on the surface of it. But we are sort of forgetting that where we started was pathologists looking down the microscope at sort of subtle cell and tissue morphology. And when we quantify single cell, this information in single cells down to sort of one number per protein per cell, we're losing a lot of that rich morphological information. Furthermore, when we actually look in we've actually done some 3D imaging, multiplex 3D imaging. This is surface rendering, thanks to Clarence, of a confocal scan of a 5-micron tissue section on the left, and this is a nuclear channel, and a 30-micron section on the right. So the classic histology section you get from the clinic that all that H&E and the 100 years of beautiful science has been done on is a 5-micron section. And we didn't think about this too much, or maybe we kind of ignored it, but 5 microns, you're not getting any single whole nucleus. So you can see each of these nuclei is just shaved across the top and the bottom. So if you're not getting a whole nucleus, you're not getting a whole cytoplasm either. Furthermore, what about the cell that was above or below? That empty space might have cytoplasm of some other cell in it. So when you go to quantify this image, if your segmentation boundary for the cell next to that sort of, half cytoplasm cell includes part of the cytoplasm, you're going to be quantifying the wrong cell type. And if it's different cell types, you got immune cell next to a tumor cell, you're going to get some weird quantification artifacts. So again, on the right, the 30-micron section, just about as thick, 30 or 35 microns. That's about as thick as we found you can mount on a slide without other things like antibody penetration and imaging quality start to suffer. But also imaging everything in confocal in 3D is also not feasible due to time and disk space. So we're looking strongly now at what we can learn from a smaller number of 3D experiments in this format. That's also thicker tissue sections with light sheet and some other technologies to figure out what we can learn and maybe bring that back to the 2D, sort of 2D, pseudo 2D image section like you would get from the image on the left. Questions about that? However, the pathologist has been dealing with that thing on the left for so long that there's still something there. So it's not hopeless. It seems hopeless, but I think it's actually fine. We just have to come up with the right way to think about the problem. So one of the, as I mentioned this a little bit, one of the problems you start to see if you start to analyze these cells as though they're 2D images and you're trying to figure out, well, if I see intensity in this pixel, it belongs to this cell, and if I can see intensity up here, it belongs to this cell, is because there's cells that are on top of each other and crowding, and you don't even maybe have a whole cell, it's sort of not a well-founded problem. So this has been, this sort of, the ramifications of this have been noticed by people in the past, and there's this algorithm called RedSea that was posted a while ago that purported to fix this in already captured images. We tried it, it doesn't work very well. I kind of don't know how it could, but on the right, you can see a representation of what this would actually look like in a real image. So the white dots indicate cells assigned to a single cluster according to a post single cell analysis clustering. The white outlines indicate the cell boundaries. The blue indicates CD8 staining, and red indicates keratin. And I'm not a biologist, but the little I do know is that CD8 and keratin don't exist in the same cells. So you've got a cytotoxic T cell in CD8 staining, and then you've got the epithelium around it in red in the keratin cells. However, because of this sort of uncertainty of where the signal is relative to your cell segmentation boundary, that maybe because of those 3D issues we can maybe never resolve, all 5 of those cells were counted as CD8 keratin double positive cells. So this is a problem with treating things at the single cell level and then trying to treat them as a bag of cells, or as some people like to say, treating the microscope like a crappy flow cytometer. Let's use the microscope for what it can do and actually use the whole texture and contour of cell images to learn from. So this is some work also by Greg Baker in the lab. Currently manuscript in preparation. I'm using convolutional variable autoencoder to learn directly on pixel images, the image pixel data, without using any segmentation at all. So the basic idea is feeding the images through into a convolutional variational autoencoder. There's one piece I missed here, which is that each of the cells, each of the small patches of the image centered on a cell is extracted from the original image and then fed to this network. It's also weighted by a Gaussian window so that the center of the cell is weighted strongly, the edges of the cell and its sort of contacts with neighbors, which may have been captured in that square vignette, are suppressed. So we're only learning in this case things about the cell itself and not its neighbors. This is fed through a variational autoencoder network, which learns basically to embed each cell in a Gaussian distributed latent space. In this case, I think they used 850 dimensional space. And the process is, the training process of this network minimizes the reconstruction loss between the input and whatever image the output images that are produced by the trained network, with the constraint that this latent space is Gaussian distributed. That has a lot of cool properties. So one thing you can do with this is you can take all of your cells from your single cell segmentation, or sorry, from your the cell points, your cell vignettes from the raw image, feed them, see where they land in the latent space, and then do clustering on that space. And this tends to work a lot better than when you cluster these single cell data, which had all these weird spillover effects of, say, CD8 keratin double positive cells. So in this case, the tumor on the left clusters and the stroma and immune cells on the right clusters separate well. The clusters make sense, and you don't have any of these strange double positive effects. So there's a lot more to this. Again, this paper, I don't want to belabor this too much, but the paper's coming out. One quick thing I'll mention is that when we talked to our pathologists and we said, okay, does what we're seeing with these, so for example, a lot of these are 1, 2, 3, 10 clusters that in the classic single cell segmentation based clustering were all identified as just adenocarcinoma. This is a colon adenocarcinoma, not surprising. But the VAE clustering in the latent space showed these 10 discrete clusters in those same cells. And they look the same, they have roughly the same intensities in the sort of single cell averaging approach. And we talked to our pathologist and we said, what are the differences between these? Can you tell anything about these from a trained pathologist's eye? And he said, oh yeah, absolutely. And went and labeled all the different cell niches, sort of tissue niches that were represented by these different clusters that you could identify, apparently the VAE could identify by just the texture of staining across all the different marker channels within the cell boundary itself, not even including neighbors. So you say, oh, well, you just got different neighborhoods. The window is small enough, and this Gaussian window sort of de-emphasize the neighboring pixels. So this is really just the texture in the cell that can tell you this fine level of detail about cellular location and tissue niches. So there's a lot more, again, a lot more to this story that I don't have time to talk about. I'm running short on time, so I'm going to skip through a bunch of things. So unfortunately, so the middle piece of my talk, which is about these visual analytics and tools to perform hypothesis generation and exploitation of the images themselves in conjunction with other single cell data, basically keeping the image as a primary artifact and not just throwing it away for scatter plots. Last thing I want to touch on is Minerva. So if we want to promote the image itself as a useful and critical data artifact, then when we go and publish papers and share data with the world, we want to be able to let them see those images. So just a tiny little thumbnail of 1 of 20 tissue samples in figure 2B of your paper is not going to cut it. So let's just share the data with everyone. Well, there's some challenges to that. One of them is technical. Fires are huge. I said they're 100 gigabytes, maybe more. We can download them on campus. We're a lot blessed with good internet connection, but if you're somewhere else in the world, not a great internet connection, that's not going to be feasible. Just holding those files is also going to be challenging. The other one is these microscopy images tend to be in specialized file formats, even if they're open file formats like OMI-TIFF, which we love. Still not something that somebody has software on their desktop that they can just double click and open. And lastly, there's technical issues where these are high dynamic range images. The brightness and contrast does not just makes it so that the intensity of the image just doesn't show up on your screen. You've got to fiddle with settings. Anyone who's opened fluorescence images in ImageJ or whatever will know how much time that takes. And now you've got, say, 30, 40, 60 channels of that to do. Other challenge is more interpretive. These are huge and complex images with a lot of biology going on. If you just dump the image in front of somebody, they don't necessarily know what they're going to be looking at. They're not going to be an expert necessarily in your disease and in your biology. So how can we navigate the viewer of the image through the biology that we want them to see? Skip through this. So the tiled pyramidal image storage enables lazy loading of just the piece of the image that somebody's looking at. So that's one key technology. I am late, so I'm going to fly through these. How we convert these sort of apparently dim, low dynamic, high dynamic range, but so the image is can be high dynamic range, but the useful part of the signal is in maybe a certain part of the histogram. We have to isolate that by brightness contrast adjustments. Then we often colorize, pseudo-color, and combine those channels into one cohesive image whole. This is what we call image rendering. Your image desktop software like Amaris, Fiji, Napari does this in its sleep. You've got plenty of data storage and bandwidth and compute locally. But if you want to do this in a web browser, the web browser is the standard way we're going to share data with the world these days. It's a lot trickier. There's bandwidth constraints, there's caching and storage constraints, and if somebody wants to look at this, say, on a phone or a weaker laptop, the compute constraints can be a problem. There's a few different approaches to solving this on the web. One of them is the Omero approach. So Omero is an open source microscopy image management tool. It solves the problem by pushing all the complexity into a server process. Somebody's got to maintain that server, care and feeding, updates, and all that, computing power. But then all the rendering happens in the server, and then it just ships a compressed JPEG to your browser, and that's that. There's another approach exemplified by a tool called Viv, developed by Nils Gellenborg's lab here at HMS. And that takes the primary image data in an open format, such as OMI-TIFF, streams the raw data in this tiled pyramid format across the web, and does all the rendering in your browser. This is great because you don't have to have any server infrastructure. You can just put the image file on a website, and this works. Downside is there's still a lot of data. So all those may be compressed, but not lossy compressed. Primary image data tiles are streaming across the web, which does take a lot of bandwidth and a lot of memory in your browser and a little bit of computation. We've developed an approach in our tool called Minerva Story that is sort of in the middle. So we do have a server process, but it's really offline. So it's run by a data collector up front. When they're going to share their data, it pre-renders all the images, all the image channels into pre-compressed image JPEGs, just like the Omero. but it does it for the whole image at once, saves it on your web server, and then we have a very lightweight web page component in JavaScript that displays those on the screen, lets you pan and zoom around the image. The downside of this is, while in Omero, the user can adjust those rendering settings to get the image just the way they like it. In Viv, you can do the same thing totally in your browser. With the Minerva approach, that has to be done up front because all those choices about dynamic range and coloring and things has to happen before these JPEG files are created. So that is one downside. I say fixed star because the one thing we did do to handle that is we created a feature where people who are sharing images can identify groups of channels that they want to present together. And those can be pre-rendered into their own JPEG so that at least the user can switch between groups of channels. We found that was usually good enough. The other key piece of Minerva that solves the second half of my dilemma, which is the interpretation, is what we call a digital docent for images. So inspired by pathology education, where you've got pathologists looking down a multi-headed microscope, navigating the slide around, telling the students what they're seeing, while the students look down the other parallel objectives to see the same thing and learn exactly what they should be looking at. We get this sort of informally from our pathologist colleagues in the lab all the time. We love hearing them narrate exactly all the features they're seeing in an image. We wanted to be able to reproduce that for people browsing my images at home. So we had this sort of inspiration of the museum audio tour. So when you're going to a museum, you want to learn about all the different pieces. You're not an art expert. Your museum provides a guided tour where you go up to an exhibit, you punch in a number on your phone or on a little headset, and you hear an expert narrate what you're looking at with context and history. You can follow the guide from point to point as the sort of museum has intended, or you can stop off, wander freely, pick up the tour wherever you like. And we thought that was a great metaphor for sort of combining the pathology education approach with these large tissue images. So what does this look like in practice? Sort of a mock-up of what this looks like in your browser. Got textual information on the left, information about the channels and sort of your legend on the upper right. Various image annotation features such as regions of interest and arrows can be added to the image to highlight critical features. We've also got a new feature that we call the lens, which lets you peek at a different image layer and move the lens around. So in this case, we've got same section H&E imaging performed after SciSif. So you can actually get sort of a window through into what the H&E, the classic H&E would look like. So again, the pathologists love this. What I can't show you in a static slide here is that if you click the forward arrow, that will step the user through different points along the tour, and it can take them to different parts in the image, different zoom levels, different channels, and give them different text to contextualize that part of the image. The workflow for creating these stories is, after data collection on the top, you load it into our tool called Minerva Author, which is sort of an authoring environment where it lets the author create these different waypoints, write the text, define the channel groups, add annotations, and sort of build the story that build the tour that tells the story they want to tell about their image. And they click a publish button. It spits out all the different JPEG and HTML files that they need to publish a story on the web. They copy that folder to the web, and they're ready to go. They can share that URL with a colleague or the public. This is a great adjunct to your paper where you want to share, again, not just that tiny little postage stamp. You can share this link in your supplementary material and have your viewers actually navigate through images. So this is just a few screenshots of what this looks like. Here's an example of an H&E image. And here's a feature where we've got an actual image embedded in the waypoint on the left and some single cell segmentation and clustering results overlaid on top of the image. We have recently implemented what we call flexible pre-rendering, where instead of rendering full color JPEGs with a series of channels pre-embedded, we actually render every channel in grayscale independently, and we move just the coloring and combining, the pseudo coloring and combining to the browser, which is super lightweight. And now this lets people, in addition to these pre-grouped channels, it lets the user turn channels on and off, add new channels that weren't there before, change colors. So this basically gets most of the way to what people wanted out of these image browsers. They weren't super happy. Nobody who is not the original creator of the image cares about adjusting the contrast as long as the person who made the story did a good job. But people do want to look at different channels in context. One last thing before I close is all the metadata behind the clinical metadata, specimen metadata imaging itself and the analysis, we've built a ontology called MITI, Minimum Information About Tissue Imaging. It's the website. So we are endeavoring to collect and release rich metadata about all of our samples at the time of publishing these stories. So I want to thank everyone here. All the folks here I've listed on the left have a direct hand in anything that I've shown today. Leadership, Laura, Sandra, and Peter at the bottom. There's many people I did not thank who provided the wonderful data and did the actual experiments to let me and my team have fun working on their data. So it's too many to thank. I'd be thanking everybody in that photo. The websites for the lab and some of the various main tools that I mentioned are on the bottom right there. I can leave that up if anybody cares. Any final questions? And 1 minute over.
Thank you, Jeremy. I think there are questions on the way and we're at time. People are free to leave. Of course, they always are. It's a free country. But we will take more questions just, let's say, for the next 5-10 minutes. But I will start with one, and that is, if people have more questions, are you still available Thursdays at noon as a general rule and at the computational forum in WAP?
Yeah, so one thing that we do as part of this computational software engineering team in LSP is what we call the computational forum. So we found that if we're going to push computational tools on people, we can't always do sort of complicated, long, drawn out training sessions, classes. So we decided to sort of split the middle, and we have kind of office hours where people come 1 day a week. It's basically me and my team sit in the room. We try to figure out what people want to do ahead of time. We send it out on a Slack channel. And then if somebody wants to learn, say, about the best image training and image segmenter, or they want to learn about how to make a Minerva story or how to do whatever, we'll send out the agenda, and then other people who might be interested in that same topic can come and listen to the discussion. So it's not just office hours, it's meant to be a little bit more of an open discussion. So it's, it's Thursdays you should probably contact me so I can tell you more and we can get you on the agenda, but it's Thursdays at 12 in Systems Biology Department in Warren Alpert.
WAP 436?
Yeah, Warren Alpert 436. But if you do want to show up, email me first so just we can get acquainted. Yes.
No. Oh well. So if you're a quant scientist, getting the cell boundaries, getting the cell segmentation, getting it to read stuff, that's vitally important. That's one of those big problems right there. But if you're a clinical pathologist, if you're just or you're an anatomist, do you still really care about that? Because as long as the stuff that's there lines up with your micromedia and protractor, as long as the structures and organs are still there, your pathology is actually quite your histology. I mean,
sure,
is quite different.
So anybody who's just looking at the image with their eyeballs, yes, the image itself, they don't even need any cell boundaries because you can see. I mean, you look by eye, you know where all the cells are. Your brain's very good at picking that up. But yeah, it's the quantification. Absolutely, it's the quantification and spatial identifying spatial patterns and relationships between cells and knowing are the immune cells excluded from the tumor? What part of the tumor is there? Immune synapse. Like there's tons of things. Yeah, we need to know very 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 precisely. We're trying to figure out where that VAE approach can sort of either maybe not replace, but maybe cross-pollinate with these other single cell level quantification approaches. We're not quite sure exactly how it's going to work, but it does seem like it has some promise.
The cell is just a lot of it seems to be on the metadata side, which we didn't have time to go over. It's just, so how you make the case, how do you do that? Is it just consistency from the readings, trying to get more consistent segmentation? Is it not so much just getting it correct, but making it the same between?
I believe there are a number of clinically approved tests that are using single cell segmentation on histology slides. So people have convinced the FDA that it's at least workable. It's at least consistent enough. So I guess the challenge is, my hope is that some of the errors and the problems I showed with the sort of you don't have a whole cell, you have the overlap. Hopefully, in the aggregate, those things are uncorrelated errors, And that's at least just gonna spread out your distribution and widen your error bars, but it's not gonna change the fundamental, your fundamental analysis, I think.
Okay, we have a question from online. I'm gonna read it. It goes from Antonios. Can you describe your efforts towards incorporation of multiplex image decoding, if any?
Multiplex image decoding?
Yeah, I think he's thinking things like fish, Merfish, things like that, where you
Oh.
decode. I'm guessing.
Yeah, so we definitely use other techniques in conjunction with SciSif, usually on an adjacent section. So a lot of these things are not compatible with IF imaging. Adjacent sections are tricky because, again, with looking at the thickness of the slide compared to the size of the cell, you're not gonna get the same cell. You're gonna get regions that are the same. So you can do sort of regionally based analyses, but it is tricky to integrate those data types across different sections. But we definitely use, for example, we use geomics, although maybe not for much longer now that NanoString's being sued out of existence, but we use that as a sort of paired assay with SciSif to gain complementary information. Yeah.
I think we're going to stop here. I have a bunch of questions, but I'm going to take them offline with you. So thank you again.
Thanks, Simon. Thanks, everyone.